Chapter 99. Heart-lung preparation. Introduction. Heart-lung preparation is an experimental setup, devised by Starling. It is used to demonstrate the effects of various factors on the activities of heart, particularly heart rate and cardiac output. This preparation is also used to record the cardiac function curves. Procedure. Heart-lung preparation is usually done in dogs. After giving anesthesia, neck of the dog is opened and a tracheal cannula is inserted into the trachea. Tracheal cannula is connected to a respiratory pump, so that respiration in the animal is controlled artificially, to avoid any disturbance during the experimental procedure, Fig 99.1. Then, chest is opened and an arterial cannula is inserted into one of the branches of aorta. All the other branches from arch of aorta and descending aorta are ligated. Arterial cannula is connected to two instruments, mercury manometer to measure the arterial blood pressure. Air bottle, which provides elasticity artificially, as in the case of arterial wall. Thus, the blood ejected from left ventricle passes into air bottle through the arterial cannula and rubber tubes. From the air bottle, the blood is diverted through a tube which provides artificial resistance. Air bottle is also connected to a pressure bottle. Pressure bottle is attached to a pressure pump. This pump is used to maintain the pressure within the setup. Artificial resistance is offered by applying pressure surrounding the resistance tube. Resistance tube is also connected to a manometer. After passing through the resistance tube, blood is allowed to flow through a warming glass coil, which is kept inside a water bath with a heater. Temperature of water bath is controlled, so that the temperature of blood could be maintained. Warming coil is connected to a venous reservoir through a flow meter, which determines the amount of blood flow, cardiac output. Venous reservoir is connected to superior vena cava by a rubber tube. A screw-type clamp is fitted to the rubber tube. This clamp is used to adjust the amount of blood returning to heart, venous return. A thermometer is also fitted to the tube to note the temperature of blood. A third mercury manometer is connected to the inferior vena cava. It is used to determine the venous pressure. A cardiometer is fitted to the ventricle. This cardiometer is connected to a recording device like Mary tambour or polygraph, to record the ventricular volume changes. Pulmonary circulation is kept intact for continuous oxygenation of blood. Uses of heart-lung preparation. Thus, in this setup, the heart works as an isolated organ. So, the effects of various factors can be demonstrated on the activities of heart, like heart rate, ventricular volume, and cardiac output. Examples. When venous return decreases, stroke volume decreases. When venous return increases, stroke volume increases. When resistance increases, cardiac output decreases. When resistance decreases, cardiac output increases. Heart-lung preparation is also used to record two types of cardiac function curves, cardiac output curves. Venous return curves. Though the cardiac function curves are obtained in experiments using the animals, these curves represent the functions of the ventricles in human heart also, Chapter 100.